Welcome to the Pancreas lecture, the Endocrine Pancreas lecture, because as we will find out if we don't know already, the pancreas is considered both an endocrine and an exocrine organ. So let's go ahead and get started. In this chapter, I'd like you to pay attention to a couple of things. First off, just normal pancreatic anatomy and function. We'll talk about the physiologic units of action, cellular subtypes, so alpha cells, beta cells. There's a weird like delta cell and X cell. And then your physiologic feedback loop here involves insulin and glucagon. It says to know what governs their release, and I can tell you already it is glycemia. So this isn't going to be a loop that involves the hypothalamus, pituitary whatsoever. It's only going to rely on blood levels of glucose. And the important thing to keep in mind is that glucose cannot enter the cells that need to utilize it unless there is insulin present. So when there is a spike in glucose in the bloodstream, you will see insulin release. To kind of tie that all together, we'll end with diabetes, which is a general pancreatic dysfunction, a very common one when we think about the pancreas. There's a type 1 and type 2. If you understand the subtypes and how they differ, then you will do just fine on the exam, and then also your boards, which will have a large subtopic section in pathology and in physiology regarding diabetes. All right, so the basic anatomy of the pancreas is that it is associated with the liver because, as we've learned previously, the liver is manufacturing bile, which will converge down this common bile duct. And in those individuals with a gallbladder, will also back up to the gallbladder, which will contract when there is a signal from CCK, cholecystokinin, which will trigger an increase in pressure and then the opening of the sphincter of Oddi, or the hepatopancreatic sphincter. And why that is important is whenever there is bile release, there's also going to be the release of pancreatic juices. And these juices are exocrine in nature, meaning that they're created by acinar cells collected in the common pancreatic duct, which will converge together with the common bile duct so that all of those secretions will be liberated into the duodenum when the CCK and secretin signal to the pancreas. So the pancreas itself has a tail and a head portion, which we'll go ahead and look at here again. So here is the general body or parenchymal tissue of the pancreas. The tail portion is more to the left side of the body, so distal from the opening of the pancreatic duct into the sphincter of Oddi, ampulla of Vater area. And the head of the pancreas, because it is most first line in contact with the duodenum, considering that the alimentary canal, the duodenum being a part of the alimentary canal, would be the closest relationship there is to any sort of irritant or piece of the external environment. Why that is important, for those of you who have listened to the other lectures, you would know that when there is damage to the pancreas, it's almost always in the head portion. And in cases where that occurs, of pancreatitis or pancreatic cancer or a stone impaction, what you see is that the common pancreatic duct becomes impeded. So flow through it is impeded, which traps the pancreatic juices within the pancreas. Why that is so dangerous is because of an exocrine product called trypsin, an enzyme that digests proteins. So unfortunately, when there is damage to the pancreas at the cellular level, it's almost always in the head portion, which does control the outflow. However, regardless of where the damage is or why the damage occurred, not only do we have to keep in mind that there is exocrine function with regards to digestion, there's also the endocrine function of the pancreas, which will be affected. So as we saw before, we have acinar cells. So these are secretory cells making exocrine products, delivering them to a duct, which will then become the pancreatic duct. In the endocrine function, we have cells of the islet, so rather than acinar cells, which are grape-like clusters of cells, we have an islet named after the eponymous individual who discovered it, so it's referred to as the islet of Langerhans, which contains the endocrine cells. And when we have endocrine products, those are released into the bloodstream, which is why you see the venous outlet here, rather than into a ductal system. In this picture, there's two types labeled, although there are additional types of cells. But we have two of the most important. We have the alpha cells, which make a product called glucagon, which acts in opposition to the beta cells, which make insulin. Glucagon and insulin do function completely opposite of one another. One is released when blood sugar is high. One is released when blood sugar is low. So looking at the physiology, again, it connects to the duodenum. We know the pancreas as a general organ can be exocrine or endocrine. The exocrine function was already discussed in the digestive portion, whereby we have the creation of multiple enzymes, lipase, trypsin, amylase, and bicarbonate, which are emptied through the duct. The endocrine products are not involved with the duct, they are involved with the bloodstream, and they include insulin and glucagon. So there's a dual function of the pancreas. As we discussed in the digestion chapter, there are a huge, very scientific term, there's a large amount of pancreatic juices or fluids that are created and then secreted per day. We also learned that one of the most important functions is the bicarbonate that's produced by the pancreas, so that when we have chyme coming from the stomach into the duodenum, the pancreas delivers a bicarbonate to neutralize those really acidic contents so that we don't have any damage or ulceration of the duodenum. And those digestive products include our trypsin, our lipase, and our amylase to digest the various macromolecular structures in food, so carbohydrates, proteins, and fats. So all of the different pancreatic juices are here, and that's just important to keep in mind as we think about the way that the pancreas interacts in both an endocrine and exocrine function with the physiology. So going into the endocrine physiology, the islet of Langerhans, again, this is going to contain our endocrine cells, which in this picture are labeled with dark blue, light blue, and purple in the center of this functional unit. We see the acinar cells here. Those make up a duct, so they're making secretory products, which are then secreted into the duct. There's about 1 million different islets in the pancreatic tissue, and these islets will converge together to make the various endocrine products associated with the regulation of primarily glycemia, or the level of glucose in the bloodstream. So in this picture, we actually see pretty much every single cell that we'll be talking about. We see some capillaries interspersed with alpha cells in dark pink or red. We have our delta cells here in yellow, and then in the center, in an orange type color, we have the beta cells. So about 25% of these cells, which is kind of evident by this picture, are the alpha cells, which are making glucagon. The remainder, the largest remainder, are the beta cells. So about 60% of the cells in the islets are beta cells, and those beta cells are responsible for the manufacturing of insulin. The interspersed yellow cells are delta cells. About 10% of them are delta cells, which make somatostatin. And somatostatin, soma is body, statin comes from the word stasis, which means location. So it is, that's not a great descriptor, somatostatin, but what it is telling you is that's something that tries to make static the body glycemia. Somatostatin is an antagonist for glucagon and insulin. Because we don't have a large I lost my words. We don't have a large axis type feedback here. We're just relying on the level of glucose in the bloodstream to trigger either the alpha or beta cells to make their products to increase or decrease glycemia. When there is nothing else needed, somatostatin will work in an antagonistic nature, turning off the functionality of those cells so that we can stop its action. There isn't an override system as we see with other axis types of endocrine tissues, so somatostatin is going to be the shutoff for this system. Finally, there's an obscure cell type, which nobody really knows for sure what it does, so you'll even see in pictures they don't even include it because nobody wants to talk about it. It's called an F cell or a PP cell, and it makes something called pancreatic polypeptide. And if you're familiar with some chemistry, polypeptide could refer to literally anything that's created in the body through transcription and translation. Because we don't really know what pancreatic polypeptide does, very obscure function that's being investigated, again, that's something that's not even labeled, but do know that they are there to some degree, and we are investigating their functionality. Okay, so the important part of the pancreatic function is understanding the relationship of glycemia to the release of insulin or glucagon. So this functionality is very straightforward. It's a physiologic feedback loop, no involvement of the brain, no involvement of the hypothalamus, the pituitary. Instead, we see only glycemia, which refers to the level of glucose in the blood, controlling the release of these various endocrine products. So taking a little look at this picture, we have low blood glucose states. So those would be fasting type states. So those could either be cases where somebody is completely controlling their carbohydrate and sugar intake through something like a ketogenic diet. Those could be fasting states. For example, when you go to sleep at night, the body still needs to work and it runs on glucose, so we still need some functionality. Or this could be the time between meals in American diet types of cases. So when there is low blood glucose, there's a signal to the pancreas, which has cells there that really just are going to monitor levels of glucose. That will turn on the alpha cells, so the alpha cells start to spit out glucagon. When glucagon is released, that will signal to cells in the liver to start glycogenolysis. So you might know that glucose is stored in the liver as glycogen. Glycogenolysis is the breaking apart of that glycogen into its respective parts so that the free glucose can be liberated into the bloodstream. That will help normalize homeostatic levels of glucose so that we have functionality of cerebral tissue, muscle tissue, and really all of the tissues that utilize glucose as an energy source. In an opposing fashion, when there's high levels of blood glucose, so that could follow a meal, especially one that's rich in carbohydrates / sugars, which are one and the same. When that occurs, that high level of blood glucose, so high or increased glycemia, signals to cells monitoring glycemia in the pancreas that it's time to produce insulin. The cells that produce insulin are called the beta cells, and when insulin is around, it encourages the tissues that can utilize and store glucose to functionally take in glucose so that it can be utilized. If there is an absence for any reason of insulin, the glucose remains free in the bloodstream, which can cause a lot of very troubling effects physiologically. This is why in diabetic states we see high levels of blood glucose, so very high levels of blood sugar, because there's no insulin to physically traffic the glucose into the cells that can utilize it. This is presented in a very functional and simplistic way with a feedback loop. High glucose states, insulin is produced by the beta cells to lower glycemia down to normal. In low glucose fasting states, the alpha cells produce glucagon to raise the glucose or glycemia level to our homeostatic normal. So they oppose one another. They work in a very antagonistic fashion to one another, and then somatostatin is the overriding control here to help turn off in an antagonistic way both of those products. This is what an insulin receptor looks like. So this is what we call a dimerized receptor. When there is the binding of something to an insulin receptor, a dimerization refers to essentially that receptor unfolding so that we have two opposing mirror image copies of it. So it's a dimer, it's a mirror image. So together it is created by 4 subunits held together by disulfide bonds. 2 are alpha, which reside outside of the cell membrane, and 2 of the beta subunits. Those are proteins that traverse the cell membrane, so they're integral proteins. Insulin itself binds to the alpha subunits, which in turn phosphorylates the beta subunits. When that phosphorylation occurs, we have the activation of tyrosine kinases so that we have the phosphorylation of insulin receptor substrates, or IRS. As we mentioned in the introductory chapter, most of these hormonal products in the endocrine chapter will involve some sort of second messenger signaling, which will involve the activation of tyrosine kinase, one of the most common second messenger systems in the body. So insulin binds the alpha subunit, the beta subunit interacts with this tyrosine kinase system so that we have the physical phosphorylation of these substrates, so that we have fat synthesis. Fat is a great place to store glucose, protein synthesis, glycogen synthesis, so that we can store glucose in the liver. Protein synthesis, you might guess, is in the muscle. Then we also have growth and gene expression associated with growth factors, IGFs, insulin-like growth factors, when there is binding of insulin to its receptor. So the mechanism of secretion of insulin is going to involve glucose receptors. So on the right, you can see a picture of a cell. We see a GLUT2 glucose receptor. And so here, listen closely for those of you who have drifted off. There are only 2 cell types that are permeable to glucose at resting potential. One is neuronal. So whenever glucose can get to the brain, those brain cells will take it. They do not care. They do not discriminate. The other cell type that is permeable to glucose at its resting temperature are the physical beta cells associated with the pancreas. Okay? So that means we can have glucose entry into the beta cells of the pancreas. Why that is important is that's how insulin is released. You first need to have glucose entry into beta cells. So we can see here this process whereby glucose is trafficked into cells. We have a physical depolarization event that turns on that cell. When there's depolarization, the end game eventually is that voltage-gated channels will open to let in calcium, and calcium drives docking and fusion of vesicles. So glucose comes in, we have second messenger kind of signaling cascade. That's the short story. And then we have resulting depolarization of the beta cell, calcium entry through voltage-gated channels, which drives the docking and fusion of insulin. So you cannot have insulin release without glucose coming in. And that makes sense. Glucose is what triggers insulin release. Insulin has no job in the body besides helping traffic glucose into cells that need it. So the GLUT2 receptor is what traffics glucose in and allows the exocytosis of insulin. All right, so that is the way we get insulin secretion. Glucose entry into beta cells and then insulin production. Again, that's important because no other cell besides brain cells can do that. There's only a minute amount of glucose that can be utilized by beta cells and by the brain. The rest of the glucose will remain free in the circulation, causing damage and causing pathology. So it's extremely important that insulin be exocytosed so that we have some utilization of glucose. You can also visualize that by looking here at this physiologic graph. On the x-axis, we have plasma glucose, so we can see it goes from 0 to 600, so those are levels of sugar in the blood. And on the y-axis, we have the amount of insulin that is secreted into the bloodstream. As you can see, at a very hard level of 100 mg per 100 mils, we start to have insulin released in result of glucose going up in the blood. So glucose in the blood, so glycemia goes up, insulin goes up. The only thing that insulin needs to do is traffic glucose into the cells that utilize it, period. This is a rapid release graph showing an animal study with glucose infusion. On the top here, we see in orange the length of the glucose infusion. On the X, we just have minutes. So what you're seeing on the Y is just levels of either glucose or insulin. So this is a big, big bolus dose, an infusion dose of glucose. Then we are measuring the level of insulin in the plasma. So insulin is released with a spike fashion, where immediately after a high carbohydrate meal, or in this case, a controlled glucose infusion, you see insulin release immediately spike. Within 10 minutes of eating a high carbohydrate meal, and the same goes with like sugary things, because sugars are considered carbohydrates. So that would involve things like soda and alcohol, which is a very, very simple sugar. So within 10 minutes, we see the levels of insulin rise, and then an immediate reduction of plasma glucose in normal individuals after a meal. This is a steady state glucose level, so we're looking here at still an insulin spike, and then insulin production goes down. And then we see a steady rise in insulin following the meal, or in this case, the infusion. So within the first 10 minutes of this spike, we have a fall. So there's a spike and a fall. This is the early phase of insulin release. However, food remains in the stomach for 1 to 3 hours. Then it's going to go to the duodenum, where it'll be fully broken down and then absorbed. So there'll be a slow, steady rise of insulin over the resulting hour to help counteract that immediate spike you might see to glucose levels in the bloodstream. So just some basics. I'm not going to read every single sentence to you. I'm going to give you the basics so that you can read them on your own. Insulin action is most important to physically cause cells to become permeable to glucose. Again, there's only 2 cell types that are permeable to glucose at their resting potential. They are the beta cells in the pancreas, which are making insulin in response to that. And then the other are cerebral neurons, which need it to run on. However, in the rest of the body, it's primarily anabolic. It helps maintain the upper levels of blood glucose, glycemia, free fatty acid levels. And what insulin does is physically move glucose into cells. Everything that you see here on this picture, muscle, adipose tissue, liver, those are places where we can physically store glucose. However, they will not let the glucose in if insulin is not there first. So this is a dual binding interaction where you need to have insulin bind for glucose transporters to open. That is important. It moves glucose. Without insulin, you will not have glucose enter muscle, adipose, or liver hepatic cells. Insulin and muscle, so muscles love glucose. At resting potential, we don't have good permeability, just slight permeability. When insulin is around, we can traffic glucose into those cells. Muscles use huge amounts of glucose. A few hours after a meal, during moderate to heavy exercise, we get a ton of glucose utilization by muscles. Insulin also encourages glycogen storage in muscles so that glucose is not hanging around in the bloodstream causing damage. It can be stored in the muscle as glycogen. And glycogen can be broken down very, very easily and quickly, and it's great for short spurts of anaerobic energy when we don't have physical oxygen and ATP cascades. So it's a great source of energy, and we need to have insulin to physically traffic it there. Insulin promotes uptake, storage, and use of glucose. Insulin causes glucose to be trafficked into liver cells so that it can be stored as glycogen. Without the presence of insulin, the liver will not do that. So it encourages enzyme activity, it encourages utilization of glucose, it encourages the activity of glycogen synthase, it encourages utilization and uptake. So it's more likely that glucose be stored by the liver and then utilized most effectively by the liver. Finally, glucose release between meals. This is something that is under the control of glycogen. When we have fasting states, insulin helps to reverse glycogen storage. Glycogen isn't synthesized. The liver takes up no more glucose. We start to activate and then allow phosphate radicals to split away from glucose, and so free glucose can enter the bloodstream. So glucose enhances utilization, storage of glucose at every level in the tissues that can physically do those activities in the anatomical body. Excess glucose, you might know, is converted into fatty acids that can be packaged as our triglycerides, as our low-density lipoproteins, so that would be considered the negative or abnormal type, or not abnormal, but the disadvantageous type of cholesterol. Insulin also inhibits gluconeogenesis, or the creation of new glucose or glycogen, by decreasing enzyme availability. So we are able to bring the glucose into the cells that need it only with the presence of insulin. Insulin, though, does not have a ton of effect on the brain because, as we've learned, there is an ability of cells in the brain to take up glucose at resting. Most cells in the brain are permeable to glucose at resting. They don't really need insulin to traffic it in. Brain cells normally only use glucose for energy and not fats. This can be forced by shifting away from a glucose-heavy diet into a more ketogenic diet. And that is something that was first established by really parents who were looking to control pediatric seizures. Neurons run on glucose. The more glucose there is, the more brain activity there is. So there was an initial idea that shifting the brain away from glucose metabolism and into a state of ketosis would limit seizures. And so for a true ketogenic diet, we go through the amounts of each kind of macronutrient in pharmacology, but it's almost 95% pure fat. So when you see people doing ketogenic diets, typically it's not that much fat. For a pure ketogenic diet, it has to be about 95% fat. Those are cases where you will see the brain utilize ketone bodies rather than glucose for an energy source. For those individuals who do diets of that nature, they talk about kind of a ketosis fog or a flu that starts in the first couple of weeks since they shift. And that involves the brain physically making the shift from a glucose energy source to a ketone energy source. It does take a little bit of energy, and it's not the preferred fuel for brain cells. But just in general, brain cells, that's what they use. They use glucose. Insulin isn't super important to neuronal activity because they are permeable to glucose at rest. They do not need the assistance of insulin. as we'll learn with diabetes, low blood glucose is something that can cause hypoglycemic shock. And hypoglycemic shock, there's a lot of nervous irritability. And when I say nervous, I mean nerves associated with the nervous system. And the nerves kind of go into this weird hyper/hypodrive kind of pendulum swing, where there will be overactivity followed by inactivity. So you'll see fainting, seizures, coma. This is a fun pharmacology fact. Before they used really heavily electroconvulsive therapy, there was a time that they would put people into diabetic shock by alterations to glucose, so that you would have the same kind of convulsive therapy that we see used now. So glucose is readily taken up by the brain. Insulin also promotes fat synthesis and storage. So if you just want to think about the ways that we store glucose, it's either stored in the liver, in the muscle, or into fat cells. So fun things about fat. So fat cells will store glucose. They love glucose. They will take it in and they'll use it in your fun biochemical cascades that you learned about, like glucose, pyruvate, acetyl-CoA, wonderful citric acid cycle stuff. But at the other non-biochemical side, when adipose cells take up glucose, they can only store so much. When they are full of glucose, they will make a copy of themselves. And so then adipose cells will split and fill up with more fat. And then they'll split again, and so on and so forth. So they love to store glucose. They'll store it as free fatty acids. They'll store it as whatever you need, but they'll take it up. It is released from the liver as lipoproteins, so those are your LDLs and your HDLs. All right, when there's insulin deficiency, you probably know that as diabetes. Insulin deficiency leads to uncontrolled level of glycemia. So what you'll see is normal dietary intake of a food, lack of insulin secretion, and then blood glucose going really, really high. The other problem with that is that it goes along with this creation of free fatty acids. So you have a high level of fats. You can go into something called acidosis, which is a overproduction of acetic acid, which will drive down the pH of the body. That is referred to as diabetic ketoacidosis. That is a life-threatening problem, and it is something that we try to prevent patients from going into. So insulin deficiency, instead of having glucose trafficked into cells that can utilize it, the glucose builds up in the blood and we have an increase in glycemia. This is just insulin and proteins. Insulin does stimulate transports of amino acids into cells, and amino acids are the building blocks of proteins. So the more insulin there is, the more transcription there is long term. It does inhibit catabolism of proteins, and it inhibits gluconeogenesis in the liver. And this is a little picture down here to show you the overall picture of insulin and proteins. But more protein synthesis, more protein storage, and prevents the breakdown or degradation of proteins. So insulin is important so that we can retain our muscle, retain our fat if you want to keep it, and also retain our stores of glycogen in the liver. And we will not form those stores of glycogen without the presence of insulin. Insulin also acts in a very synergistic way with growth hormone. So this is a really old school animal physiology study. On the Y, you're looking at weight, and on the X, you're looking at days. So this is depancreatized, meaning there was a surgery to remove the entire pancreas, and hypophysectomize, which means the hypophysis, which we've already learned is the pituitary. So they took out their pituitary, they took out their pancreas, and then they started giving them things. All right, how fun. So that's how science works. Like, all right, let's take out the natural way that those things are produced and let's give it artificially so that we're controlling all of those control measures and measure just one thing. So they took out those things, they allowed the animals to heal, so they've got no pituitary, they have no pancreas. And then they supplemented hormones. So because there's no pituitary, the researchers supplemented growth hormone for about 50 days. And what you see when you look at this graph is that there wasn't a lot of growth. We know that growth hormone promotes long bone growth and anabolic activity. We do not see that. Then they supplemented insulin. Insulin didn't have any change on weight. So these animals were not making their own growth hormone or insulin. These were delivered by the researchers for an extended period of time. Next, what the researchers did was mix together or deliver both together, growth hormone and insulin. And you can see that when that started, there was a pretty steep peak in weight in those animals. So what is hypothesized after this study was that insulin and growth hormone work in a synergistic way to promote growth. When something is synergistic, it means that they get together and they promote the activity of one another. So by themselves, there's not a huge indication that there's a change in weight. However, when they're given together, they have a synergistic function where they both increase the activity of the other hormone. Fun times, so that works together. All right, the end stuff here, we talked about insulin, so the pancreas makes insulin, and the opponent of insulin is glucagon. Glucagon is your hyperglycemic hormone. It is a physical antagonist to insulin. So insulin lowers blood sugar by trafficking glucose into the cells that can utilize it. Instead, what glucagon does is raise blood sugar levels by liberating stored glucose, which is stored as glycogen, in its sources. So when glucagon is released, glycogenolysis goes up, so glycogen is broken down, and increased gluconeogenesis, so new creation of glucagon so that we can continue to tap those stores. Glucagon is a member of the secretin family. We talked about secretin in the digestive chapter. It circulates unbound to proteins. It has a half-life of about 6 minutes. It has a very strong first-pass effect, meaning that the liver extracts glucagon really readily when it's released. And there's 2 different types of amino acid sequences in something called pre-pro-glucagon, the very pro hormone of glucagon, GLP-1 and GLP-2, which are now. So because excuse me. So because we have glucagon being secreted to raise blood glucose levels, to increase glycemia, what you see is the opposite of what you were looking at with insulin graphs. So when blood glucose starts to drop, if we look here at the y-axis, we see that glucagon expression is high in states of low blood glucose, whereas you start to approach these upper levels of glycemia, and we see decreased glucagon secretion. So essentially, glucagon is going to be released in an antagonistic way to insulin. Insulin is around to lower blood sugar, whereas glucagon has the primary role of increasing available glucose to the body. And the way that it does that, if we take a look here at this feedback loop from your textbook, one of the textbooks, we see insulin and beta cell production, and I want you to notice just these little feedback loops where it says inhibit secretion over here. So these 2 products, insulin and glucagon, inhibit the other's secretion. So when insulin is around to decrease blood glucose, it will also turn off alpha cell secretion of glucagon, and vice versa. When glucagon is secreted, then we are going to have an inhibitory action with beta cell secretion. Instead of seeing the inhibition by insulin itself, one of the main things that you'll see for a drop in insulin secretion, so glucagon isn't going to physically inhibit much of the beta cell activity. Instead, what you will see is circulating catecholamines, so your dopamine, epinephrine, norepinephrine, will stimulate the beta cells to turn off and inhibit their secretory activity. And also we have the local somatostatin, which is being secreted by the pancreas. So stimulus for release of glucagon is really when you get into situations where there is a drop in blood glucose. So I've already mentioned things like fasting states. So that would be the first thing you have up there, drop in blood glucose due to time between meals. This also happens after exhaustive exercise, where you start to break down some of the muscle. You've utilized any blood circulating levels of glucose in exhaustive types of exercise. So we start to see glucagon activity increasing the liberation of the stored glucose in places like the adipose tissue, the muscle, and the liver. Also, when there are a presence of serum amino acids, and amino acids get together to make peptide strings, which become proteins. So after a really high protein meal, alanine and arginine are two of the most prevalent amino acids that will stimulate release of glucagon by the body. So again, we've got circulating catecholamines shutting off these systems when necessary, and somatostatin playing a role in that as well. On this slide, what you're looking at is just glucagon binding to its receptor, and through a variety of intracellular signaling cascades is able to increase the utilization of alternative or stored fuel sources. So at the end of the day, there's going to be increased glycogenolysis, so breaking down those stored glycogen to liberate glucose, activates the adipose cell lipase to start breaking down the fats, oxidizing free fatty acids, and we'll see increased gluconeogenesis so that we have a nice store. And this is going to liberate our fuel sources so that they're readily available. Okay, as I mentioned before, just a quick little note, the catecholamines, they're all here for you, dopamine, epinephrine, norepinephrine. They directly bind to beta receptors, which are prevalent on muscle, adipose tissue, and liver. You should know that catecholamines also bind to alpha receptors in some cases, but in the counter-regulatory phase for glycemia, we're worried about beta receptor binding. Catecholamines are released in any time the body enters a state of homeostatic stress. So anything that's set by the hypothalamus, so caloric expenditure, exercise, temperature abnormalities, any of those things that we consider homeostatic, or in times of psychological stress as well, so that we have a sympathetic response. And you would expect catecholamines to be released with a sympathetic response to initiate the sympathetic response. So when they are around, we are going to inhibit things like insulin activity, because we don't want to lower the blood sugar when the body is stressed or when the body is exercising. The glucose needs to be available to the exercising tissues. Just a final note tying everything together with diabetes. Diabetes mellitus is a disorder. There's two different types. There's type 1 and there's type 2. And primary type 1 diabetes is an abnormality of the beta cells. There is some sort of autoimmune component to this. It's not very well understood, but there is immune destruction of the beta cells in the pancreas. And so in type 1 diabetes, there's an inability of the beta cells to physically make insulin, and then there's uncontrolled levels of blood sugar. Type 2 diabetes is normal pancreatic activity, and there can either be a insensitivity to insulin by receptors, so that we have receptors that don't respond to levels of glucose and levels of insulin the way that we would expect. So in the same way that your sensory system can undergo adaptation, like with smell, you quickly start to lose the ability to recognize that you're wearing a perfume, for example, because that's a redundant signal. So the body just becomes attuned to it and starts to ignore that response, that sensory response. So we can have the same type of thing happen in type 2 diabetes, where insulin receptors don't respond to insulin. Or there can be a case where simply the body outgrows the pancreas. The pancreas can only make so much insulin. So if the BMI is very high, there has to be a supplementation of medications to help control glycemia. So at the end of the day, it's really beta cell dysfunction. Either there's going to be an abnormal or lack of insulin production, or an insensitivity of the tissues to bind insulin. If insulin is not around to move glucose and shuttle it into the cells that can store it and utilize it, then it remains in the bloodstream where it can cause quite a bit of damage and a lot of very profound types of pathologies that we talk about in pathology. This is just explaining type 2 diabetes. It can, again, either be sufficient insulin, but it's not binding and being recognized by the receptor subtypes, or it could be the outgrowing of the body to the amount of insulin that can be manufactured by the pancreas. And that ends pancreas. Here is your sample board question. It should seem easy now. The following hormones released by your pancreas to lower blood glucose: dopamine, epinephrine, glucagon, or insulin. Always go with your best answer. Sometimes there might be something that kind of trips you up because it could be right. I want you to concentrate on choosing the best answer instead of going down that rabbit hole. And with that, we are done.